Welcome to the deep dive of Key to My Research, the science podcast by the Excellence Strategy at the University of Tübingen. In this episode, we will take a closer look at the research of Professor Eric Kemen. For the past 6 years, he has been at the Interfaculty Institute of Microbiology and Infection Medicine in Tübingen, focusing his research on microbial interactions in plant ecosystems. Welcome, Eric. We're very glad to have you here today.
Thanks a lot for this great introduction.
Yes, you're welcome. So, Eric, as a professor for microbial interactions in plant ecosystems, what is it you do?
So what we are doing is, in my group, we are particularly interested in interaction between microbes, but not every microbe. We are interested in microbes that live on plants, that live on and in plants. And we are studying there from ecological aspects, or how do these microbes behave in the environment, down to molecular mechanisms. So how does it really work that microbes talk to each other, interact with each other, and particularly also how do they fight with each other to basically defend their habitat where they are living.
So we'll get back to your science later. Now we'd just like to learn a little bit more about you. So tell us about your history. How did you get here where you are today?
So basically I started early on when I was at elementary school with microscopy. And I was early on very much interested how it looks like what you don't see with your eyes. And later on, I basically started with agriculture. I went to an agricultural school, did agriculture there and learned how to basically run a farm. And from there, I wanted to know how do really plants and microbes interact. So I started to study biology at the University of Constance, did also my PhD there. And from there, I basically went to UK, where there was a really excellent center for plant sciences in Norwich. And this is where I got really into contact also with bioinformatics and more the molecular mechanisms of the interaction between the microbes. And from there, I got then the opportunity to become a group leader at the Max Planck Institute in Cologne. And this was the first time where I could really develop my own ideas, my own group, and where I could learn how to run a group and to do my own research on microbe interactions, because that has just come up at that time as a very important topic. And one has just realized how important it is that not only one microbe interacts with the plant like this was seen before, where one just looked at pathogens as a single pathogen on a plant, but one realized it's far more complex, so there are far more organisms. And this I could develop in Cologne. And after some years there, then I got the opportunity to take up a professorship at the University of Tübingen, where I then could start my group, where I'm currently developing the ideas of how microbes interact, but also, of course, how these interactions with the plant and the plant itself make the plant basically robust, so that it is robust to environmental factors with its microorganisms together and the plant on its own.
Mm-hmm. So let's go back a few years. You mentioned wanting to become a farmer initially. What farmer would have you become?
Well, so I would have probably become a more organic farming person. So I was always interested in alternative farming, so where you basically don't use a lot of agrochemical, but where you basically use more the natural resources that exist in order to build up your farm. So that would have been my idea. But of course, it's not easy if you don't come from a farm to become a farmer and to get land to do this actually. So this then had to be an idea and stayed very theoretical.
Okay, but you basically stayed in a similar business. You're in agriculture, you're doing research for agriculture. And what is it that basically drives you to do this kind of research?
So, of course, what drives me is I'm really interested in basic research where we understand how the molecular mechanisms work and how this works in ecology. But of course, this should be somehow applied. So we are looking into this with always the idea, how can one use this in agriculture later on? So how, for example, can one get a plant to survive much better under very harsh conditions? And I think that's a very interesting aspect to consider when one considers agriculture. So how much efforts we have to take for the plant to survive in the field. And if you just look around, how many plants just grow wherever, in forest, meadow, wherever you have such a diversity and they just grow. So there are some pathogens around, but not so much. And even if, as we had it now over the last few weeks where there was a lot of flooding, even a meadow usually survives this flooding fairly well. While if you have crops like maize and it's flooded, so usually the plants start to die. And this is something I was always very much fascinated. So how do these wild plants with all their complex environment survive all these stresses? Why our farming plants basically are so sensitive. This is, of course, due to monoculture, but it doesn't have to be monoculture all the time. And of course, one has to combine this and come up with new agricultural ideas. And this is basically where we want to go. So we want to get an idea on the molecular mechanisms, and we really want to show how one can apply this, for example, in organic farming later on.
Mm-hmm. So talking about harsh environmental fluctuations, climate change, what is it your research can do to help make these plants more robust? So how do you go about it?
So we have different strategies. So one strategy is, of course, to start with the plant itself. So we first have to see why are particularly wild plants more robust than the agricultural plant. And this has to do, of course, with breeding. So they have been mainly bred for yield, but one hasn't looked very much that they become robust or stay robust like the wild ones. And there the important thing is basically to become a better understanding of how can we get back this robustness to agricultural plant while still maintaining the yield, of course, because one has to be aware that we have to feed more and more people on the earth. So basically we cannot really risk that we are producing less. Also the plants are more robust. So we really have to see that we get these plants robust by learning something from the nature. And this is where we start with the plant. And one thing is, of course, to look at the genome. So we are very much interested to look at the genes that are relevant for this. And this is also what my colleagues are doing at this plant institute where we are in Tübingen. And our main goal in general is to figure out what does it mean for a plant to become robust. So what does it mean if you stand out there and you suddenly have sun that burns down like hell, and basically their temperature increases to 60 degrees because the ground gets hot, to 70 degrees, and the plant has to stay. And then during the night it suddenly drops, or in winter it drops really below zero and so on. But still the plant basically stays. And we have not really a good idea what this robustness is and how we can basically change it or adapt it. Or, as I said before, so one important thing is currently that it changes rapidly from drought, where there is not enough water, particularly in the east of Germany. We have still this problem. Also this year that there's not enough water and the plants basically get very dry and they have to resist. And then suddenly there's this lots of rain coming and then it floods for days, right? So there's suddenly water standing and then the oxygen levels go down and so on, and the plant suffers a lot. And also there it is important to understand how can we get the plants robust to this harsh condition. So how can we avoid that with these conditions that are more and more coming to the global chain, that we basically enforce our plants to be more sustainable in the way and in the conditions we have.
Mm-hmm. So which regions are you looking at? Because climate and the weather and the sunlight is obviously different depending on what region you live in. So where's your focus?
So currently our main focus where we do the basic research is really in Germany. So currently we are mainly looking into plant systems really around Tübingen and on the Swabian Alp, because the Swabian Alp is an extremely harsh condition for plants. So this is probably you know that there long ago people were only able to grow lentils, which was one of the main crops there, because this can really survive harsh conditions, which is also interesting as a plant itself. And of course, we are looking at also wild plants on the Swabian Alp and around Tübingen and so on. And we try to get an idea how these plants basically survive. But of course, we also want what we get at basic knowledge. We also want to get further, for example, towards the global south. So there we are collaborating with groups in Madagascar, because Madagascar has a broad range of climates, so really from tropical to more Mediterranean dry habitats. And within this area, we are, for example, studying rice. So because rice also, first of all, it needs a lot of water. But of course, if the water levels get too high, there are other problems like toxicity we are looking at with iron that suddenly becomes available in too high concentration and then becomes toxic. And then suddenly the farmer is not able to harvest any more rice. And this is, of course, a problem, particularly in countries where the farmer really relies on what the yield is for the family to survive. And this is where we want to apply it. So this is more our applied research, but the basic research we mainly do here in Germany.
Mm-hmm. So do you have a typical day as a scientist at the University of Tübingen? How would that look like?
A typical day. So usually I get in the morning after I've brought my kids to school, so at 8. And then when I start in the lab, the first thing is to look at new papers and so on. So what is basically out, new literature. So I try to screen what has come out. So what have colleagues published? Is there anything new? And then I would basically go to the lab and check with the people. So what has gone on during the last days? What has been happening? Are there new results and so on? And then usually, so as a professor, you also have a lot of administrative stuff, unfortunately. So the nice part is the science. And when I have seen the science, I usually then have to go to my administrative jobs.
So I'd really like to learn more about the real-world applications. Could you elaborate a bit and tell us about the timeline of your research? Because, of course, climate change is a very relevant and pressing topic at the moment. How quick can you be with your research?
Well, of course, that depends very much on the methods we have available. So, of course, if you do this with classic breeding and you want to get really the results we have from the lab into the plant, this will take a very long time. So what we generally using also in our experiments are, of course, transgenic plants. So we are mainly knocking out genes or bringing genes in also from wild plants. And there we, of course, then also need to study in the field, or would like to study in the field, how these plants really behave. But of course, the regulations in Germany are very strict, so we are very limited in what we can do. But luckily, so this has been changing over the last years gradually, and the acceptance has become higher, because one just has to be aware that it's very difficult to really breed in all these genes with classical breeding in the real time that we need with these changes, because we are already seeing that we have very harsh conditions for our plants that, I mean, most people have really faced already this year and will face also during the summer, really extreme temperature, extreme hot, extreme water. And of course, there the plants need to adapt, and we need to find a way how to really get this to the agriculture very quickly. And of course, one has to say, here, of course, we can import if our yield just fails. But of course, that's not possible in all countries of the world. So one really has to think of how one can basically do this to get our basic findings into the plants and help the plants to survive much better.
Mm-hmm. And what mechanisms do you use, or what methods do you use to extract genes and put them in different plants? Could you elaborate a bit on the methods you use?
So for the methods, basically, if we want to get a gene out, there are very old methods like PCR techniques, where you just amplify this out. And there are very modern techniques like the CRISPR-Cas technique, where you bring genes into other genome, or where you also can basically knock out genes if they are a problem for the plant and they are not ideal under harsh conditions. And one has to say this has nothing to do anymore with the techniques that have been used in early days. So if we have a plant that would go to the field, there are, of course, no antibiotic resistances anymore in this and so on. So the techniques have changed so much that the risk of carrying something over is extremely low, or basically zero, that we don't know and that we don't want to carry over. So therefore, I consider this a very safe technique to really generate transgenic plants now, and to use the knowledge we have from our basic research to really make the plants more robust.
Mm-hmm. So depending on the region of the world, people often have a different attitude towards genetically modified foods. And of course, there are lots of critics. There are people that think it's an innovative, future scenario. How do people react to your research?
Well, generally, they are very open to the research and they are very much interested in how one could basically apply this as well. But it's very controversial, of course, if one can use transgenic plants in agriculture or not. And as I said already, in Germany, that is a very difficult debate currently. And but one has to say, if we don't want to stay with our classical farming, or conventional farming, where we use a lot of agrochemicals, we have to consider other ways. And particularly if we want to go to organic farming, which I already said in the beginning, I really favor and I was very much interested in, one has to make sure that also there the tools are available to have the same yield. And of course, there the plants need to be particularly robust, particularly if they grow together with other plants. They need to compete with other plants. They are facing a lot more microbes that are coming in, pathogens and so on. So they really need to be more robust than in the conventional farming. But therefore, we are really integrating it in more natural system. We are keeping the diversity up and so on. So I think that is really a benefit for the nature. But of course, if we really want to make this sustainable, we need robust plants, and the breeding there often takes far too long.
How would you explain genetical engineering to someone that's not from your field of research?
Well, genetic engineering is whenever you start manipulating a genome of an organism. And this can be in two ways. This can be you start deleting genes, or you insert something into this genome of the organisms.
How dangerous is this?
Well, in my opinion, this is not dangerous because we are just making small changes to the genome. And compared to how this was when people started with GMOs, where they brought really huge cassettes into the genome with resistances to antibiotics and so on, we can do this now really precise. So you can basically not distinguish anymore if that was a natural mutation that happened over time or if that was really introduced. Of course, we are documenting all this and we are not selling something that we made as non-GMO. But this is currently a big debate because you don't really see the difference. If you wouldn't know this comes from a lab, there are no differences at all to natural selection. And usually if you have crops standing out there in the field, they mutate much faster. So you have much more diversity that gets just due to the UV light out there and all radiation and so on. So this is much more diversity that happens usually naturally than what we introduce into such a plant to give it new properties.
And in Mother Nature, how long would the changes take that you do in the laboratory?
Well, if you want to do this for agriculture, it usually takes 10 to 20 years until you have a good crop with the new properties. Because if you just cross two, or you even cross in wild plants, which is possible, then you have a huge mixture suddenly, because you mix the whole genomes with each other. And then you have to select all those plants again for the property you are really interested in. In this case, what the example was for resistant gene. Of course, if you just cross the plants, you're not just crossing in the resistant gene, but you're crossing in a lot of genes in the background as well. And you then have to see that you get rid of all these genes in the background again, or at least you need to get them to a status where they don't influence, for example, your yield or other properties. So that is why it takes so long, because you need many generations to screen until all this, what happens in the background, is basically corrected again, and is really then a variety you can actually use in agriculture.
Mm-hmm. And what are the beneficial traits that make a plant more robust?
So robustness, that is, of course, what we want to figure out. So that is our current research. So we are very much interested in what makes actually the plant robust. I mean, we know that there are resistant genes that make it robust to pathogen. There are genes that detoxify certain metabolites that accumulate when the plant is growing underwater and so on. But this is what we are interested with our current research. So we want to figure out what really makes the plant resist. So what is it? Which genes are actually there? And how do they play together? Because often it's not just a single gene, it's much more complicated what's happening in such a plant. And we really need to figure out what makes a plant robust.
Mm-hmm. But can you understand the critics or the reservations that people might have, especially in Germany? Where do you think this comes from?
Yeah, this comes from the early days of genetic techniques and genetic modification, where particularly genes had been introduced that made the use of particular agricultural chemicals better. So one could use more agricultural chemicals, and the farming plant basically that one brought to the field was more robust. And this was probably a mistake, because people didn't really see what it benefits them. So it was basically for particularly on the commercial side. So it didn't really help the individual people, and it didn't really help to keep the diversity up. So it was really just to make conventional farming more sustainable. But what we are really talking about is keeping the diversity thanks to these techniques. So, and not just using these techniques to make more money than we have done before and ruin basically the diversity. And that was a big mistake they did in the early days, where it was basically just used to make more money out of it. But it was not considered as a help for the farmer, particularly to really get the diversity up in the fields. It was rather the way to get it down. And of course, there was also this problem that was always discussed, that at that time, due to the screening techniques, how they brought in the genes, there were often resistant genes to antibiotics left within the plant. So that does not matter too much, because the plant is mainly resistant anyway. And it did not spread to other plants or did not spread to microbes or whatever. But of course, it was a strange feeling that these antibiotic-resistant genes were also within this plant. And of course, this was not good that they did not explain this properly to the public at that time.
Mm. Talking about the public, what do you as a scientist think it would need to bring more acceptance for your field of research, or generally for genetically engineered plants and crops?
Well, I think the most important thing is to talk openly and to show what we are doing, and that this is basically no secret. So that what we are doing is nothing magic or whatever. It should be really shown more to the public how this works and what the benefits are as well. So that we really can generate with these plants where one would basically not see that this gene was brought in with techniques in the lab, but was not basically bred in. So the problem with breeding is usually you mix a lot of genes. So there you have much more risk that you mix genes that you don't want to mix, and then you have to select very long. And particularly this selection process then for the right plant takes extremely long and takes often too long, particularly if it comes to pathogens. Then the pathogen can evolve very quickly and can overcome these resistances that one has just bred in. And then, of course, it takes a long time to get, again, new crosses ready and so on. So if we want to be really fast and be more successful with it and really avoid chemicals, so avoid all the fungicides, herbicides, we need really robust plants. And I think this robust plant we can only get if we bring in the targeted gene or delete the gene that is probably not favorable for the plant.
Mm-hmm. You were saying that you take a look at wild plants, Mother Nature, and what can these plants bring into our crops? Like, what is it they do which our crops can't?
Well, so they are often really well adapted for the habitats where we want to grow our crops, right? So they grow there for a very long time, and our crops are often brought in from other countries. Maize comes from South America, and this is, of course, not that well adapted to our environment here. And what we can learn is, so maize is also grass, so we can basically look at other grasses that are around, and grasses are very well adapted here. And we can learn from those ones how they basically survive and how they can adapt to this. As I said, as one example is flooding. So we know meadows can be flooded for a very long time, which often happens. They are often growing close to rivers, and they are very used to flooding. While our crops, and this is not only maize, this is also wheat, barley, and so on, they are not very robust to this flooding. So one thing would be then to look into why are these wild grasses so robust? So what do they have to survive this, and why do our crops don't have this anymore? So why was this basically bred away, or did it not exist perhaps in the beginning? So don't we have varieties that are really robust to this? But one important thing is also not only go to the wild plants, as you mentioned, but we also need to better look into the pool we have. So, of course, there are a lot of old varieties that have been grown and which were not grown anymore due to they were not as efficient in the yield, or they were not as good for mechanical harvesting and so on. And we also should go back to those collections and look into what basically is available there in terms of diversity. What can we use there? And how can we get this basically into our crops?
Mm-hmm. And what sort of old sorts are you specifically looking at?
So we are looking into, of course, barley, wheat, and so on. So for maize, that is more difficult. We are looking into those ones, but we are also looking into Fabaceae, like I said, for the lentil. So these are all these plants, so this whole group. Fabaceae is basically all these beans and so on. And there, my group particularly is looking into lotus, which is Fabaceae, which is closely related to lentils, beans, and so on. So all these plants. And Lotus corniculatus is one of those plants that you basically now find everywhere. So it's a yellow flowering plant which becomes almost like a small shrub. And this is extremely resistant to environmental conditions. It also survives over several years. And of course, from such plants, we can really learn how the survival strategies are. So we can learn this, of course, from the genome itself, but we are also very much interested in how microbes interact with this plant and how this plant is basically getting robust through the microbes that it has. For example, for Fabaceae, it's very common that they are interacting with bacteria that can fix nitrogen on their own. And in agriculture, of course, nitrogen is always limiting. So the farmers have to bring to the field a lot of nitrogen fertilizer. And those Fabaceae plants, they are basically able, together with the bacteria, to get their own nitrogen from the air, because our air contains 80% of nitrogen, and they can use this nitrogen in order to fix it, and make it available for the plant and produce amino acids and so on that they bring in. So this is also what lentils do. And this is why they are so well adapted on these harsh conditions, for example, on the Swabian Alp, where these elements like nitrogen are very limiting. And thanks to this interaction with the microbes, they can basically survive there and make their own nitrogen available for themselves.
Mm-hmm. So from the Swabian Alps to the beautiful city of Tübingen, you've been here since 2017. What makes Tübingen especially good university for you in your research?
Well, one thing, of course, why I came here and chose to come here from Cologne before, was that here the plant research is really a focus. So there we are in one big institute where we are only looking at plant research. And we also are associated with ecology, for example, that also has a big part looking into plant. And this is really a center for plant science. And of course, this is a big advantage if you have colleagues nearby that all work on plants and all different aspects. And this is, I think, one of the unique sides where you have such a focus that so many groups work with the same interest on the same topic. And for me, it was also interesting to come here and very important because it's not only plant sciences, but we also have a very strong microbiology. And within this microbiology, the microbiome is one of the hot topics and the main topics they are focusing on. And as I'm part also of the microbiology, this is making the perfect environment where one can study on one side the plant, one can study the microbes, and one can really bring this together with the ecology to study how plants interact with their environment to really survive these harsh conditions.
Mm-hmm. And looking into the future, what goals do you want to reach with your research?
Well, of course, the main goal would be that what we find can really be translated into agriculture. So this is, of course, the goal. This is also why we are collaborating very closely with Hohenheim. Hohenheim is the main agricultural university here in the south of Germany. And it's nearby, basically, which makes it an ideal situation that we have this basic research in Tübingen and we have the applied research really in Hohenheim, where there are also the fields available to do all these studies and the big greenhouses. And this makes this really a unique site. So we have the opportunity to develop something and to study in detail, and then go towards the translation. And also in Hohenheim, they have then the connection to the farmers to really reach out and bring this to the farmers in order to be tested. And this is really the goal where we want to go. So we really want to understand from the gene, from the individual microbes and its genes to the applied field, that in the end the farmer can really use it and that this becomes basically a product on the market that really helps the people to feed the world in the end.
That's a big goal.
Well, that's a very big goal, and I can certainly work on this until my retirement.
That sounds good. Other than being a unique site for your academic research and for reaching big goals, what do you like about Tübingen and how do you spend your free time when you're not in the laboratory or on the field or on the Swabian Alps?
Well, what I really like about Tübingen is first of all, it's an extremely international city. So it's a very small city. And usually small cities in Germany are not so international, but Tübingen is completely different. So it's an extremely international place, and this also changes the way of life, how it is here. So it's a really beautiful city with a lot of opportunities to go. There's a lot of cultural events and so on. And for me personally, I think what is important is that it's really embedded in natural reserve, like the Schönbuch, which is a huge forest area which is really natural, and it's really fantastic to go out there and spend days, because you can just walk from Tübingen outside and you can walk for hours and hours and hours just through nature.
And when you're walking through nature, do you always have the perspective of a scientist?
Well, I don't know if that is of a scientist, but we are, the whole family, my kids and my wife, we are very much interested in plants in general. So we are very fascinated by the diversity that is out there. And of course, we want to know which plants are there if we don't know them, and try to identify them together. And of course, we are also, since I'm also into microbiology, we are very fascinated by the diseases the plants have. This might sound a bit strange, but there's such a diversity of microbes you can basically see if you look closer at the plant. So there are, for example, rust fungi. So they live on the plant, they leave the plant alive, and still they can grow very well on those plants, and they make fantastic structures where they release their spores in the environment. And you can see them on the plant. They are very colorful. So there are orange spores, red spores, brown spores, and so on. And this we as a family like very much also to look into those pathogens. Of course, what you find always are a lot of mildews that are outside there. And this I find also very interesting, to just look at this diversity, so that just seeing by naked eye how fantastic the diversity is that is out there. And the fantastic thing is also you have all these pathogens there. The plants are still all very healthy. So they have some pathogens, some leaves are infected, but the plant overall is healthy and can grow very well. So, and that is, of course, fascinating, looking out there in nature. And of course, what we usually have with us is a small pocket microscope. So we usually also try to look closer into insects and whatever we see there, and how the structure also look on the microscopical level, to get an idea what we really find out. Because these are just fantastic structures if you look in the microscope, and it's so fascinating what nature is generating there.
Okay, so typically I wouldn't take a microscope on walks to relax in the woods. So this isn't a typical walk.
Perhaps in Tübingen it's known because we have so many people studying biology, and I'm sure a lot of them are interested in plants. But if you really want to see the fantastic details, I mean, just take a microscope and just look at the plant, and you will see how fantastic the structures are that you are not seeing with your naked eye. So it's just fantastic what you are seeing. So I think that's particularly for the kids very impressive usually, and they like this a lot and like to do this as well. But not only the kids like it, also the students usually like it. And we are also offering student courses. And within these courses, we usually have a lot more people that register than we can actually take. We also go out in the nature and show them also which diversity they are finding there. And I'm sure that a lot of the students, when they afterwards go on walks, they do the similar things, and they are also impressed by the diversity they are seeing. And it's very difficult, if you have been once doing this science and looking into this diversity, to see the world different anymore. So, right, you cannot just walk through green and consider it as green.
Okay, I won't do this anymore, and I'll definitely take the microscope with me on my next walk.
Perfect.
Good. So I'd just like to talk about the outcomes of your research. Are there any concrete outcomes that we can talk about?
So our outcomes in Tübingen are mainly basic research. So we know genes that are relevant, and of course we want to know more of those ones. And of course we are collaborating with Hohenheim to get this into the field, but currently the acceptance for transgenic organisms, if we want to bring these genes really in, is very low, and in Germany it's just not allowed. So therefore we don't have really field studies where we can show this. So on the other hand, my group is working particularly with microbes. And of course, there more and more acceptance is coming to use probiotics. Probably everyone knows probiotics, so you have it in your yogurt and so on, so you know it. But there you often have monocultures. Perhaps they then advertise that there are 3 bacteria, 4 bacteria, But in reality, such a plant lives with much more microbes together. And of course, these microbes are very beneficial for the plant because they can protect it from pathogens or can also help with nutrition. And this is what we are interested in. So we are really looking into which microbial communities are associated with this. And we study how these microbial communities are really building up. And what our hypothesis is, so you don't have to bring all the genes in because a lot of the genes are also within the microbes that live with the plant. So as the example I mentioned, that, for example, nitrogen fixation can be done by microbes and they are associated with the plant, and then you don't need the fertilizer anymore because the plant can get from the bacterium the nitrogen with which it lives. And of course, these are things we are very much interested in to develop and help farmers at one point to say how can they basically promote the microbial communities that favor their plants. And this is, of course, in conventional farming, this would be very interesting to increase the diversity, to really get out more microbes into this field, or go to organic farming where we basically increase this diversity on purpose. And this has been done for a very long time already. People know that if they bring a lot of organic material into the soil, the microbial community increases. But also if you spray certain mixtures on the plant, you can increase the diversity there. And this can protect the plant, certainly.
And are these microbial communities, are they a real alternative to doing research on transgenic crops and engineering?
No, it's only an add-on. So what we need for sure is we also need to be able to bring genes into the plant also in order to interact with the microbes, because these interactions are very specific quite often. So there needs to be also something on the plant side to get into this interaction and on the microbial side. And therefore, it would be ideal if we could just get genes into the plant to go into interaction, or directly for the plant to become more robust and defend itself against pathogen or harsh environmental conditions. But as the acceptance is very difficult at the moment, this needs to still some work that basically one explains to people what the advantages are and how dangerous it is or not dangerous at all, that one can basically bring in these genes into plants to protect themselves and to become more robust.
Mm-hmm. And like talking about climate change, I mean, on the basis of what predictions can you do your research? Because it isn't sure how the climate is going to develop.
No. So, well, we know that the overall temperature gradually increases, but particularly here in Germany, it leads to far more fluctuations. And I think this everyone is already facing. So we know that there will be more extremes. So we don't know when they are coming and to which strength they are coming, but we know they are coming. We have all seen this, from fires in summer that even happen now in Germany to flooding that we have in summer or in spring and so on, and harsh winds, haily storms that injure the plants and so on. And of course, we don't know where it is going, but we know that these fluctuations get more, and therefore the plant needs to get more robust. So if we still want to get enough yield that we can really harvest enough from our agriculture, we need to be able to really modify the plants in a way that they can really survive. And there are different strategies, like using the microbes or bringing in the genes, and they really have to go hand in hand with each other. And of course, I can well understand that there is a lot of criticism and people argue there is already so much diversity in nature that one could just cross in. The problem is that until you really have the perfect crosses, it can take 10 or 20 years, and this is often too long to really become the crop that you want and that you can really use, because now we already have the problems. Now we have the flooding, we have the heat, and we need to see that we get our nutrition keeping up. Of course, we can just buy things from outside. If it fails, like this year, there will be a lot of maize that basically fails and so on, and we just buy it from outside. But of course, this then just increases the price for all these crops on the market worldwide, and then the poor countries are not able to buy it anymore. And of course, it's not fair that basically we can then buy it and get our nutrition still because our agriculture fails, while in other countries people die due to hunger and malnutrition because they can just not afford it anymore. So we really have to think what our responsibility globally is on this.
So is that also your motivation for doing the research that you do?
Yeah, that's also my motivation, of course. So it is really important to find ways to increase the yields, or to keep it at least at the level we are currently at, with all these environmental fluctuations that we are getting. So we really need to see that our crops get really more robust, that they can really survive whatever happens. We don't know where it is going to. We don't know how it will be developing in Europe, if the temperature increases, decreases, or if there is much more fluctuation. But we know that the conditions just get harsh. And these harsh conditions are just bad for plants. And this is what we are seeing currently and already facing a lot. So we have to do something now.
Talking about time, you said in 20 years it will be too late to have these results from your research. What would be a realistic or even good timeline to have these results and crops?
Well, so I hope that still in 20 years we find new things because we have gradually to adapt. But of course, to have it in crops, we have to consider, I think it's the timeline within the next 10 years that we really have to find what makes the plants robust. So what makes them really survive in nature. And then we have to get this as quickly as possible into the crops that they become really robust.
In order to reach this goal within the timeline you just said, what do you need as a scientist?
So, of course, what we need is we need the opportunity to also test our plants. So, of course, if we generate transgenic plants and we bring genes in or delete genes, we need the possibility to test this in the field. And these are two ways we need it. So there is one legal frame, which in Germany currently doesn't exist. So hopefully it's going the right way that it might exist soon. But currently it doesn't exist as such. And of course, we need the acceptance by the public. So one thing is the legal thing, but it doesn't help us if in the end the fields are destroyed. And of course, if the fields are destroyed, I don't see the problem by the people that destroy it, but that means that we have not reached everyone to really explain why we do this, why it is really needed, and why this is important for really getting along and getting people enough nutrition all over the globe. And that in Germany we have a responsibility to really do this. So I can well understand the criticism that currently exists, but I think we have to somehow communicate and find a way to overcome these problems.
Mm-hmm. Eric, I think this was a very inspirational and interesting talk, and thank you for opening up about your science. One last question. What would you say is the key to your research?
Well, key to my research is for sure to understand from an ecological point of view, so to really understand where plants are growing, how they are growing, what are the environmental factors, and to go down from these ecological aspects to the individual genes and how they function. So that is also what motivates me all the time to do my work. It's really that the way of understanding how on the really small scale we can understand how this basically translates later on in an ecological or agricultural context.
Thank you, Eric, for having you here and giving us insights into your research. This was the deep dive of the Key to My Research podcast, produced by Changing Time in cooperation with the University of Tübingen. You can find more information and the links to the sources in the show notes below. If you enjoyed this episode, please subscribe to the podcast, leave a review, and recommend it to your friends and colleagues. Thanks for listening. My name is Jennifer, and see you next time.